Hello students. Today's online lecture is on the topic gastrointestinal motility. Gastrointestinal tract motility involves the movement of ingested food through the gut. The movement is accomplished by the coordinating contractions and relaxations of the smooth muscles in the various parts of the gut like that of esophagus, stomach, small and large intestine. In today's lecture, we will see the motility of stomach, small intestine and large intestine. First, you will understand the basic physiology of gastric motility. Then, the secretions, the functions and the motility of small intestine. And lastly, you will understand the secretions, functions and the motility of large intestine. Now, let us first understand the physiology of gastric motility. Gastric motility is the function of gastric musculature, that is, the muscles of the stomach, which consists of three layers of smooth muscle fibers, namely an outer longitudinal layer, a middle circular layer, and an inner oblique layer. The motor functions of the stomach served by the gastric motility are storage of food, that is, by the process of receptive relaxation, mixing of food, that is peristalsis and retropulsion, and gastric emptying. Now, before getting to know about all the said gastric functions, you must recall a little bit about how the gastrointestinal smooth muscle functions and what are its unique properties. The main characteristic of gastrointestinal smooth muscle due to which gut motility occurs is the presence of BER, that is basic electrical rhythm. The musculature of stomach and gastrointestinal tract in general is made up of a single unit smooth muscle that has its own myogenic tone, that is, the ability to self-contract and relax. This inherent myogenic tone is due to the basal electrical rhythm. These gastric slow waves are caused by the pacemaker cells known as interstitial cells of Cajal located near the fundus of the stomach. The gastric slow waves or BER consists of an upstroke and a plateau phase. Here in this diagram, you can see the basal electrical rhythm recorded from various parts of the stomach. You can notice that the rate and force of contractions of the basal electrical rhythm increases as we go from the fundus towards the pylorus, that is, from the proximal part towards the distal part of the stomach. The electrophysiological basis of basal electrical rhythm is not entirely known, but it is assumed that the upstroke is due to the flow of sodium and calcium ions into the smooth muscle cell. Especially the influx of calcium ions is responsible for the upstroke, and efflux of potassium ions is responsible for repolarization of the smooth muscle cell. In stomach, the neurotransmitter acetylcholine increases the contractile activity, that is, it augments the action of basal electrical rhythm, and adrenaline, atropine and enterogastrone decreases the contractile activity of the stomach. Now that you know the mechanism of generation of basal electrical rhythm, let us see the various types of movements that occurs in stomach both when it is filled with meal and when it is empty. Movements present in the stomach when it is empty are migrating motor complex and hunger contractions, and movements seen in the stomach when it is full with food are receptive relaxation, mixing peristaltic waves and gastric emptying. Receptive relaxation is the mechanism by which stomach receives and accommodates the food that you eat. It is a vasovagal reflex which is initiated by the distension of the stomach, and it is synchronized with primary esophageal peristalsis. So, as soon as the entry of food stimulates the oral region stretch receptors, a signal is sent to the lower esophageal sphincter to relax and open so that food can pass easily into the stomach. Removal of vagal nerve or vagus nerve will abolish the receptive relaxation. The second type of gastric motility that is seen when the stomach is full is mixing peristaltic waves, also called as peristalsis and retropulsion movements. You must understand that the presence of food in the caudal region of stomach will generate increased contractile movements in that part, and these are nothing but the mixing movements that helps to mix the food with stomach acid and breaks the larger pieces of food into smaller pieces to form chyme. Peristalsis in stomach is produced by the periodic changes in the basal electrical rhythm of the stomach musculature. It is the coordinated smooth muscle contraction followed by a wave of relaxation, and it begins in the mid-stomach region and proceeds caudally, that is, towards the pylorus. Thus, the deeper peristaltic waves towards the pylorus are called as antral systole. Here in this diagram, you can see that the peristaltic contractions that begins in the mid-stomach region pushes the food towards the pylorus. But this is not that simple as it sounds. Actually, there occurs a kind of competition between the pushed food and the peristaltic waves to reach the pylorus. Thus, when the food reaches the pylorus due to the peristaltic push, the wave of peristalsis also reaches the pylorus before the food, and it forcefully contracts the pyloric sphincter. And then the food strikes against this closed pyloric sphincter, and most of the food is forced back again into the body of the stomach. So this is called as retropulsion. Therefore, stomach peristalsis and retropulsion makes sure that all the gastric contents are thoroughly mixed with gastric juice and grinded to a fine paste, that is chyme, before it can be emptied into the small intestine. The third type of meal-related gastric motility is gastric emptying. Gastric emptying means the movement of chyme from the stomach via the pyloric sphincter into the duodenum. It occurs due to a progressive wave of forceful contraction that includes the antrum, pylorus, and proximal duodenum to act as a single unit. Gastric emptying usually occurs 2 to 3 hours after having a meal, when the chyme is fine enough to pass through the pyloric sphincter. The regurgitation of the chyme or the backflow is prevented by the strong contractions of the pyloric sphincter, as well as due to the release of certain gastrointestinal hormones like cholecystokinin and secretin. Now let us see the gastric movements that occur when the stomach is empty. The most important type of movement seen at this phase is migrating motor complexes. Migrating motor complexes, or MMC, is the peristaltic wave that begins in the esophagus and travels through the entire gastrointestinal tract during the interdigestive period, that is, when the active digestion is not taking place. The main functions of MMC is to remove any left out food particles in the gastrointestinal tract. These contractions occur every 60 to 90 minutes during the interdigestive period and are initiated by the release of a gastrointestinal hormone called as motility. The next type of movements that occur during the interdigestive period are hunger contractions. It is believed that migrating motor complexes are responsible for the initiation of hunger contractions. Here, mild peristaltic contractions that occur in an empty stomach increases in intensity over a period of time and produces hunger contractions. These contractions, as the name suggests, are also known as sensation of hunger and can even be painful if food is not eaten on time. Now let us come to the next topic, physiology of small intestine. Small intestine is the major site of digestion and absorption of carbohydrates, fats, and proteins in the body. It is the small intestine only, especially the duodenum, where the stomach contents mixes with the intestinal bile and pancreatic juice. After mixing of the chyme, most of the essential and required nutrients are absorbed by the body at this level, and the unabsorbed nutrients and fluids pass forward towards the large intestine. Small intestine is a 6 to 7 meter long convoluted tube extending from the pylorus to the ileocecal valve, where it joins the cecum, that is the first part of the large intestine. Small intestine can be divided into three parts, namely duodenum, jejunum and ileum. Here, this diagram will help you to understand better the functional anatomy of small intestine. Here you can see a C-shaped structure present in between the liver and the pancreas. This is the first part of the small intestine called as duodenum. The duodenum is 25 cm in length and it is the widest and the most fixed part of the small intestine. The superior part of the duodenum is called as duodenal cap, and this part is mostly struck first by the acidic gastric contents, so it is a common site for peptic ulcer. The bile and the pancreatic duct opens at the level of the duodenum and releases their contents into it. The second part of the small intestine, jejunum, and the third part, ileum, form the proximal 2/5 and the distal 3/5 parts of the small intestine respectively. There is no sharp demarcation present between the two parts, only the inner mucosal surfaces can be used to differentiate between them. Histologically, the wall of small intestine is made up of four layers which from inside to outside consist of a mucosal layer, submucosal layer, a smooth muscle coat and the serosal layer. Although the small intestine is about 6 meter long, but it has an absorptive area of over 250 meter squares. This large surface is created by the special structure of the mucosal layer of the small intestine, and these special features are presence of plica circularis. These are the numerous folds of the mucous membrane layer of the small intestine. Also called as volvulae conniventis, these are the permanent folds and helps in increasing the surface area for the absorption of nutrients and also slows down the passage of gastrointestinal contents via the small intestine to help proper absorption of nutrients. The second special structure that is seen in the small intestine is the presence of millions of villi. Villi are the finger-like projections of the mucosal layer of the small intestine. Here you can see the finger-like projections. These are the villi present on the mucosal surface of the entire small intestine. Each villus is covered by a single layer of columnar epithelial cells called as enterocyte. During the process of digestion and absorption, these villi contract quickly in an irregular rhythm and relax slowly. Therefore, by this action, villi acts as a pump and it pumps the lymph towards the submucosal lacteals from the deeper core. This lymph comes in contact with the absorbed nutrients and causes effective absorption of the nutrients. Now, let us briefly see the composition and functions of intestinal juice. The intestinal juice means the secretions of both small intestine and large intestine. Also known by the name of succus entericus, the intestinal juice is alkaline in nature and have a pH between 7.5 to 8.6. It comprises of 99.5% of water and 0.5% of solids. The solids which are present in succus entericus can be divided into organic substances and inorganic substances. The organic substances that are present in the intestinal juice are mainly enzymes, namely proteolytic enzymes, amylolytic enzymes and lipolytic enzymes. The proteolytic enzymes like peptidases, they help in the digestion of proteins. The amylolytic enzymes like sucrase, maltase, lactase, etc. helps in the digestion of starch and carbohydrates, and the lipolytic enzymes like lipase helps in the digestion of fats. Apart from enzymes, there are other components like mucus and intrinsic factor which is secreted by the parietal cells of the stomach. The inorganic substances are sodium, calcium, potassium, bicarbonate, chloride, phosphate and sulfate that is present in the succus entericus. The small intestine serves some very important functions in the human body. It has mechanical functions like it mixes the chyme with various digestive juices. Small intestine serves the digestive functions also because the succus entericus or the intestinal juice contains various enzymes that digest the various components present in the diet like protein, carbohydrate and fats. Small intestine, by virtue of its special mucosal arrangements, helps in proper and efficient absorption of the digested nutrients. Small intestine secretes certain hormones like androgesterone, secretin and cholecystokinin that helps in regulating the gastric motility and secretions of the gastrointestinal juices. Small intestinal cells secrete an enzyme called as enterokinase that converts the inactive trypsinogen into active form. This trypsin is very essential for protein digestion and also for the activation of other intestinal enzymes. The mucus secreted by the small intestinal glands protects the intestinal walls from the excoriating action of the acidic chyme. Lastly, the small intestinal secretions contain lots of water that helps in proper conduct of all the hydrolytic processes required for digestion. Now that you are well aware of the physiological anatomy and functions of the small intestine, let us try to understand various movements that occur in the small intestine during the digestive as well as the interdigestive period. Overall movements of the small intestine can be categorized into motility during the interdigestive period, where the major type of movement seen is the motor migrating complex. And the type of motility seen during the digestive period, these are mixing movements like segmentation contractions and pendular movements. Then the propulsive movements like peristaltic contractions and peristaltic rush, and the movements of the villi. About the migrating motor complexes, I have already discussed in the previous slides. So let us see the various types of movements that occur in the small intestine during the digestive period. The first type of movement is mixing movements. These movements, just like in stomach, helps in breaking the larger particles into smaller ones and also helps in mixing the chyme thoroughly with the intestinal juices. The mixing movements are of two types, namely segmentation contractions and pendular movements. The segmentation contractions are the most common type of intestinal contractions that occur when chyme is present in the small intestine. These movements occur in a rhythmic fashion and also called as rhythmic segmentation contractions. Let us understand the mechanism of segmentation contractions. Here in this diagram you can see that during the segmentation contraction, a section of the small intestine first contracts and it sends the chyme in both the directions. Then the same section or the segment of the small intestine relaxes and thus the contents move back into the same segment. Simultaneously, the adjoining segment that was relaxed now contracts. Thus, this alternate contraction and relaxation of the segments of the small intestine gives a ring-like appearance that resembles a chain of sausage. The rate of segmentation contraction is about 12 contractions per minute in the duodenum and about 8 contractions per minute in the ileum. That means the segmentation contractions are more in the duodenum compared to that of the ileum. The segmentation contractions of small intestine are initiated whenever a stimulus like presence of chyme or neurotransmitter acetylcholine produces a spike potential. The frequency of contractions, that is how many times the small intestine smooth muscle will contract, is decided by the pacemaker cells of the small intestine. And the strength of contraction, that is how powerful will be the contraction, is decided by the amplitude of the slow waves. The second type of mixing movements that occur in the small intestine are pendular movements. Pendular movements are small constrictive waves that sweep the gut in all the directions in a pendular fashion. These movements also, just like segmentation movements, helps in mixing the chyme with the digestive juices. Now, the second type and one of the major type of movements that can be observed in the small intestine during the digestive period is peristalsis. Peristalsis is a type of propulsive movement that mainly propels or pushes the intestinal contents towards the anal end. It also helps in digestion and absorption of nutrients, as different nutrients are absorbed at different segments of small intestine, and peristalsis helps take the nutrients to that particular segment. The peristaltic contractions or peristalsis can be defined as a wave of contraction that is followed by a wave of relaxation of smooth muscle fibers of the intestine. Here, by this diagram, you will understand better how peristalsis occurs. Whenever the bolus of food or chyme is present in the gut, a wave of contraction will occur just behind the bolus, and also a simultaneous wave of relaxation will occur just in front of the bolus. That will lead to forward propulsion of the chyme. Now, it is not that a single peristaltic contraction occurs when chyme is present in the small intestine. Rather, several of these peristaltic contractions occur simultaneously along the length of the intestine that propels the chyme towards the aboral end. And these group of peristaltic contractions are referred as vermicular movements. The peristaltic movements at the level of small intestine moves the chyme at a rate of 1 centimeter per minute, which is a very slow rate. Sometimes, when the intestinal mucosa is intensely irritated by certain toxins or infective agents, strong peristaltic contractions occur in the gut that sweeps the contents of the small intestine rapidly into the large intestine. These powerful contractions are called as peristaltic rush. Now, the last topic for today's lecture is physiology of large intestine. The large intestine is a muscular tube of about 6 centimeter in diameter and 100 centimeter in length, extending from the ileocecal valve to the anus. It is divided into cecum, which is having the appendix, an ascending colon, the transverse colon, then comes the descending colon, sigmoid colon, then the rectum, and ultimately the anal canal. In this diagram, you can see the various parts and divisions of the large intestine. As you can see, the large intestine arises from the ileocecal junction, which is guarded by a valve called as ileocecal valve. The function of this valve is to prevent the backflow of chyme from the large intestine into the small intestine. Adjacent to the cecum and ileocecal junction, you can see a worm-shaped structure arising. This is the appendix, which is a vestigial organ, that is, it does not have any important function in human beings. Then the ascending colon extends upwards from the cecum up to the liver, and then it bends towards the left side to form the right hepatic flexure. Then the colon continues as the transverse colon, and after reaching the spleen, again it bends to form the left splenic flexure and continues as the descending colon. Now this descending colon continues as the sigmoid colon, and after entering the pelvic inlet, it joins the rectum. The rectum descends further and becomes the anal canal, and the anal canal opens to the exterior via the anus. The opening of anal canal is guarded by two sphincters called as internal and external anal sphincters. The histological structure of large intestine is similar to that of the general features of the small intestine, while the mucosa of large intestine have some special characteristic features that must be known. The mucosa of large intestine does not contain folds like that of small intestine. It also lacks the villi. The large intestinal mucosa contains large number of goblet cells that secretes mucus. Large intestine has an unusual longitudinal muscle coat that forms the tinea coli and the haustrations. The longitudinal smooth muscle fibers of the large intestine are arranged in the form of three long bands called as tinea coli, and this causes typical saculations on the wall of the large intestine called as haustrations. This is the tinea coli, and these are the saculations caused by the tinea coli known as the haustrations. Large intestine also contains peritoneal bags of fat that projects from the large intestinal serosa known as the appendices epiploicae. The large intestinal juice or secretion is a watery fluid with a pH of 8. It mainly comprises of mucus produced by the goblet cells and some water and lots of bicarbonates. The large intestinal juice contains around 99.5% of water and 0.5% of solids. The organic substances present in large intestinal juice are albumin, globulin, mucin, urea, and debris of epithelial cells. The inorganic substances present in the juice are sodium, calcium, potassium, bicarbonates, chloride, phosphate, and sulfate. The largely produced mucus lubricates the fecal matter, protects the mucous membrane of large intestine from the mechanical or chemical injuries, and also helps in neutralizing the acids formed by the colonic bacteria. Like other parts of gastrointestinal tract, the motility of large intestine is also coordinated by the basal electrical rhythm. The slow wave activity gradually increases as we go from ileocecal valve towards the sigmoid colon. That means the contractions are more at the sigmoid colon level compared to the ileocecal valve level or the ascending colon level. The two main functions of the large intestinal motility are to increase the water and electrolyte absorption from the gut and to promote excretion of fecal matter from the colon. The three main movements of large intestine are haustral shuttling, peristalsis, and mass movements. Haustral shuttling or haustral contractions are similar to the segmentation contractions of small intestine that vigorously mix the contents of colon and by exposing more of the contents to the mucosa facilitates absorption. The contraction of circular and longitudinal smooth muscles of the large intestine causes haustral contractions. These contractions last up to 60 seconds. The main function of haustral contractions are mixing the fecal matter and propulsion of the fecal matter towards the anus. The next type of movement seen in large intestine is peristalsis. As you already know that peristaltic contraction is a progressive contractile wave followed by a wave of relaxation, and these mainly helps in propulsion of intestinal contents towards the rectum. The third type of movements seen in large intestine are called as mass movements. Mass movements are special type of peristaltic contractions that are exclusively seen in the colon. These movements occur every 3 to 4 times a day, usually after meals, and forces the fecal matter rapidly down the colon. Mass movements also forces the fecal matter into the rectum and initiates defecation reflex, a reflex that causes final elimination of feces from the anus. There is a reflex that involves the large intestine called as gastrocolic reflex. It is the contraction of colon induced by the entry of food into the stomach. The reflex is responsible for generating an urge to defecate after having a meal. In adult humans, the bowel training suppresses this reflex, but due to underdeveloped neurons and lack of bowel training in children, gastrocolic reflex is very active and defecation or going to the toilet just after eating is a rule. So, this is all for today's online lecture. Thank you.